will be further discussing about some of the more important techniques which were developed over a period of time, as the basic fundamentals of the biotechnology was laid by the discovery of the restriction enzymes, then subsequently the recombinant DNA technology. Now, in the year 1975 to 81, there were various blotting techniques which were developed over there, which helped the scientists and the labs to detect the specific DNA samples or the RNA, specific RNA in the gels or the proteins in the gel. Now, the blotting technique which uses or detects the specific sequence among the DNA fragment is referred as the southern blotting, while the detection of specific RNA in the gels is referred as the northern blotting over there, while the detection of the proteins in the gel is called as the western blotting over there. Now, I will be discussing more about how exactly these particular blotting techniques helps in the detection. Now, most of these blotting techniques follow very simple procedural rule in which there is a cassette which has got the gel present within them. These gels are basically made up of micro beads which is present, through which the samples moves out. Depending upon their sizes, it moves in. So the smaller fragments moves further while the larger fragment remains on the top near the wells of the gel cassettes. Once the electrodes are being switched on, so what happens is it start moving depending upon their charge. We know that the DNA and the RNA molecules being negatively charged, so they move towards the positively charged. Once they start moving in, there is a tracking die which is being loaded. So this tracking die helps to check how far they have moved in. Once the movement has been done, the electrodes or the power supply is switched off. Now, next comes is the transfer of these fragments or the separated fragments onto the nitrocellulose membrane. This takes place through a blotting technique in which the nitrocellulose membrane and various blotting papers are being loaded into a cassette, and then subsequently these fragments are transferred into the nitrocellulose membrane. Now, once they move into the nitrocellulose membrane, they can easily detect it with the help of either the radioactive probes or the fluorescent probes. Now, we come into the first blotting technique, which is referred as the southern blotting. Now, it helps in the detection of a given sequence within the DNA. Suppose we need to detect a specific DNA sequence within them, sample. So what is done is the samples are being run over there, and subsequently the electrodes are switched on. Depending upon the sample size, they move. So we know that the smaller DNA fragment will move further while the larger DNA fragment will remain on the top. Subsequently, they are transferred onto the nitrocellulose membrane with the help of the blotting technique, and once the nitrocellulose membrane is ready, the probes are added. Now, these probes are specific for that particular given sequence which we need to detect. Now, they will go and bind to these particular detecting points over there. So, if we see the diagram over there, this is an autoradiograph which has been developed. So, this particular probe is having a radioactivity molecule over there, and it will go and detect those particular sequence or the segments over there only. Now, the next blotting technique is called as the northern blotting technique, which detects the specific mRNAs over there. Now, these particular blotting techniques detect the specific mRNA. So, what we do is from the cell, the mRNA samples are taken, and then the restriction enzyme digestion is being done. Once the samples are ready, then they are run onto the gels over there, and they are loaded onto the sample wells. So, they move over there, and the movement again occurs depending upon the sizes. So, the smaller fragments move further while the larger fragment remains on the top. Once it is being done, then it is transferred onto the nitrocellulose membrane, and in the nitrocellulose membrane, once it is transferred, then the labeled probes are being added. So, the probes again can be radioactively labeled or they can be fluorescently probed over there. So, what happens is subsequently these particular detected specific mRNAs are detected. So we can see over there, there is an autoradiograph which has been developed. So in this autoradiograph we can see there are four fragments, two fragments in each of the wells is detected, which means that each of these well or the samples contain the specific mRNA which we wanted to detect. So this plays a very important role as a diagnostic tool for the detection of specific mRNA within the samples or the patient samples. The next important technique which is there is called as the Western blotting. Now this particular Western blotting technique is being used for the detection of the proteins in the sample. Now the sample are first loaded onto the gel. Now these gels are specialized gel, many times referred as the SDS page gel, which means sodium dodecyl sulfate page gels they are called. Now what the SDS does is they give a negative charge to the proteins molecule. So they will be moving towards the positive poles over there. Now once the movement is done, we know that the proteins again they are separated based on their sizes. So once the size difference is being done, then they are transferred onto the nitrocellulose membrane again by the blotting technique and using the cassettes over there. Once the nitrocellulose membrane is ready, then they are plotted with the labeled probes over there. Now these labeled probes will go and bind to the specific DNA fragments over there or the sequences over there, and once the the protein sequences over there, so once the protein sequences are being detected, they can be easily quantified with the help of the autoradiograph, or if the probes are fluorescently labeled, they can be easily checked with the help of the machines, advanced machines. And we can see over here a autoradiograph is being shown over there. So we can see on the left hand side corner there is a ladder which has been run, which gives you an idea how much kilodalton protein is. So we can see the protein sizes over there, and on the right hand side there are four wells which are there, in which in three of the wells the proteins is very prominent, which means that the protein which is shown over here is present in three of the samples over there. So this particular technique helps to detect the specific proteins within the samples, and the subsequent other technique which we just discussed, the southern blotting which is for the DNA, the northern blotting which is for the specific mRNA, and the western blotting which is for the proteins, helps the scientists to discover and detect these molecules. Now subsequently in the year 1977 to 80, there were sequencing techniques which were developed. These sequencing techniques were basically based on the DNA replication process, because it is very important to understand what is the sequence of the DNA which has been isolated or which we need to detect. Now once the sequence we know, we can easily map out the chromosomes and we can easily detect the various chromosomal aberration, deletion, insertion, detection in these chromosomes. So it makes the things much more easier for the screening processes. Now two of the scientists, Gilbert and Sanger, they developed the different sequencing process. One was called as the chemical modification method and the other one was called as the dideoxy chain termination method over there. Now over here if we see, the DNA replication process or the method was used. Now let's see the animation over here. So we can see over here, one after another, the nucleotide which is corresponding to that particular strand, which is the linear strand which is present, comes and binds over there. So we know the base pairing. So it says that the A will bind with T, G will bind with the C over there. So one after another the nucleotides comes and there is a formation of a hydrogen bond and the base pairing occurs by the release of the phosphate from the incoming nucleotides over there. So there is a phosphodiester bond which is being formed, which forms the backbone, and then the base pairing occurs over there. So the basic principle of the DNA replication concept was used in the DNA sequencing. Now what the scientists did is they knew that whenever there is a presence of deoxyribonucleic acid, so they can easily form a base pairing. But when there is a dideoxynucleotides present, the bond cannot be formed because one of the hydroxyl group is missing, which is forming a very important role in the formation of the phosphodiester bond in the backbone. So what they did is they made 4 chambers over there. Now in the 4 chambers, in each of these chambers, the different DNTPs were loaded. Now in the first chamber they loaded, suppose the DNTP, which is ATP. In the next one they loaded DDCTP, the third one they loaded DDGTP, and the fourth one they loaded DDCTP, like this one, along with the normal DNA templates, the normal DNTPs, the polymerase and everything. Now whenever we know that in the sequence, whenever there will be addition of the DNTPs over there, which is the deoxyribonucleic acid protein segments over there, the chain termination will occur because the subsequent nucleotide cannot form a phosphodiester bond over there. So what will happen? The chain termination occurs and fragments will be formed. So in each of the well or the sample, there will be fragments which will be of different sizes over there, and when we run in the gel, we can easily make out the sequence in which the DNA is present, the DNA sequence is present over there. So this basic technique was used and it was called as the chain termination method or the Sanger method. So it laid the foundation of the sequencing or the quantification of these sequencing techniques over there. Now subsequently the various biotechnology techniques were developed which gave rise to the various other forms. So till now we discussed about the electrophoresis mechanisms, the restriction enzyme mechanisms, the antibody structures and the ELISA over there, the DNA structure and the DNA sequencing. Subsequently we will, in the next later part, we will discuss about the DNA fingerprinting, the polymerase chain reaction and the microarrays over there. Now we come into the next important discovery which was there in the biotechnology, which was called as the DNA fingerprinting or DNA typing. So we know that each of the person has got a segment of a DNA sample which varies. So there is a unique pattern of these segments which is many times referred as the mini satellites, which is present in each of the person. So we know that suppose we see this chromosome over there. In this chromosome we just focus it into one of these segments over there. So we can see that one person will having a repeated sequence which is present in 4 copy number, one person may have in 10 copy number, one person may have in 14 or 15 copy number. So each of these regions are many a times referred as the VNTRs or variable number of tandem repeats. So these VNTR regions are polymorphic in nature, which means that there are many forms which is present in different organisms. So each of the organisms is having a specific sequence of these VNTRs and they differ or they distinguish from the other organisms in the presence of the number of repeats within this VNTR region. So this particular DNA fingerprinting was used in the forensic science field actually. Now if we know that in the forensic field many a times the samples are being taken or the blood samples are being taken for that particular area in which some kind of crime activity has happened. So from the blood samples the DNA of the culprit is isolated. Now then it is done is there is a restriction enzyme mapping which is being done. So we know that there are VNTR regions which are specific, and then suppose there are 3 individuals who can be the tentative culprits who may be engaged in that particular crime area. The DNA samples from these 3 individuals are taken. They are also mapped out and they are run simultaneously in the gel. Now, if we see over here, the individual A and C, the fragments doesn't matches with the sample which has been collected from the forensic site area, while the individual B sample matches with the forensic site area. So we can easily say that individual B is the one who is being involved in these particular crime activity, which has been the blood sample which has been found in the forensic site area. So the VNTR plays a very important role, or the DNA fingerprinting plays a very important role in quantifying and distinguishing these particular areas or the sites. Now, subsequently in the 1987, another important technique which was developed, which is many a times referred as the polymerase chain reaction. Now, the polymerase chain reaction, as the name is suggesting, it is forming the polymers or a number of copies of a particular DNA fragments over there. Now, for this particular pioneering work, Carey Mullis got a Nobel Prize in the year 1993. Now, again this polymerase chain reaction works on the basic fundamental principle of DNA replication over there. Now, in the DNA replication, we know that we require a DNA template strand, which is a single stranded strand. Then there are primers, which can be forward and reverse primers. Then there is a DNA polymerase which is present, and the dNTPs which is present, which can be dATP, dCTP, dGTP and dTTP over there. Now, we know that one after another the dNTPs comes and they form a hydrogen bond, or they pair with the complementary strand which is present over there. And subsequently the phosphate which is present, it forms a phosphodiester bond over there. Now, this basic technique was used to amplify a fragment of DNA. Suppose we have got a small fragment of DNA which has been isolated from a bacterial system, and we want to increase the copy number of this particular fragments, because if we run into any kind of other techniques, it requires a larger quantity of that particular fragment. So it has to be amplified or replicated. So it is being done with the help of the technique which is called as the polymerase chain reaction. Now, the polymerase chain reaction works on the basic principle in which the step 1 gives you the denaturation process, then the step 2 is the annealing, and the step 3 is the elongation process. In the denaturation process, what is being done is high temperature are being maintained. Because of the high temperature, what happens is both the strands of the double stranded DNA, it gets separated over there. Now, the double stranded DNA gets separated. Once the double stranded DNA gets separated, what is being done is there is an annealing which happens, which means that a primer comes and binds to this particular separated strand over there. So there are different primers, which can be the forward primer and the reverse primer over there, which comes and binds to these particular segments of DNA specific at that particular point, and subsequently the third step comes, which is called as the extension point, in which a temperature of 72 degrees Celsius is maintained. Now, what happens is, as the primer has bound to that particular segment, the DNA polymerase comes and binds to that segment, and the NTPs comes and attaches one after another depending upon the complementary sequence over there. So the polymerase chain reaction helps the scientists to amplify a small fragment of DNA into a higher copy number. So it played a very important role in the biotechnology process, and subsequently the polymerase chain reaction helped for the detection of the various enzyme conditions and various protein conditions. Now, if we see over here, this is a animation which is showing the polymerase chain reaction. So if we see this animation, there are 3 steps which are being shown over here. The first step is the denaturation. So we can see the double-stranded DNA has got separated. Then the annealing happens, in which the primer gets bound, and finally the extension happens, in which the polymerase gets bound to this primer loaded side over there, and subsequently the polymerase binds and moves in with the addition of the complementary sequence of the nucleotides over there, and the DNA gets elongated. So from one DNA double helix structure, we get 2 double helix structure, in which one will be the parental structure and one will be the new daughter replicate structure template which will be formed over there. Now, subsequently, the recombinant DNA technology was used in the development of the various vaccines also. One of the very important vaccine which I will touch upon is the vaccine for the hepatitis vaccine over there. Now, the hepatitis B virus we know is a very lethal virus, and whenever this virus infects a person, what happens is, whenever a virus enters into the body, what happens is the plasma cells recognizes these viruses and they start producing antibodies for these particular viruses, which can then subsequently neutralize these viruses. Now, using this technique, the scientists developed the recombinant DNA technology to develop the recombinant vaccine over there, using the same technology of recombination, in which the specific sequence of the hepatitis B virus was used, and then subsequently it was transferred with the help of the cloning vector into a system, developing the recombinant forms of the hepatitis antigen, which then can be used as the vaccine. So whenever this vaccine, which is having the recombinant form or a truncated virus form over there, they are injected, the person develops the plasma cells, and subsequently if there is any kind of hepatitis B infection within the person, it can be easily neutralized over there. So with this note, I come to the end of the lecture, and in this part of the lecture I have discussed about the other techniques of the biotechnology, which includes the DNA fingerprinting, the polymerase chain reaction, how it was developed, and subsequently I discussed about the development of a vaccine, how the vaccines technology are utilizing all these recombinant forms of the things into their structuring based on their priorities, which includes the restriction enzymes, the recombination DNA technology, the electroporations, and finally the purification and the formation of these vaccines and the drugs. So thank you very much for your viewing. Thank you.